Hello, today we're going to show how to use GENAVi for gene expression normalization and differential expression analysis. Basically, this Shiny app, uh, it contains the first tab called gene expression, in which we're going to upload our raw count matrix. That matrix is going to pass through several normalization methods, and then we're going to use that for either visualization or differential expression analysis. So GENAVi comes with some data already pre-uploaded, but we're going to use in this tutorial our own data. In this folder in Google Docs GENAVi example files, we have some example of data. We're going to download this one, CCLE ovarian cancer RNA-seq counts. And then you choose to upload data. So we expect a CSV file, and the first column would be either gene symbol or ensemble ID, and the next columns would be only the gene expression raw counts. So it's going to upload. It's fast because I'm running locally, but you can also run GENAVi through a Shiny web server, which might be a little bit slow depending on the machine. We have a demo running on Shiny Apps.io, which might be a little bit slow since the instance we just have like 1 gigabyte of memory. So just to show that file has gene ID in the first column as expected. We have some metadata for this gene. We try to treat that in GENAVi. In case there is already metadata we do not include, but suppose we don't have it, we're going to include data from gene code. So GENAVi accepts also human and mouse matrix. The metadata we're going to add for human and the mouse are the AG38 and MM10, which are the last version available. So right now you can select which normalization method you want to take a look. You can easily export that table by clicking download and select which data type you want to export. It's going to export only the table that's being rendered. If you want the whole table, you can select all here and then just download the table. You can hide or show some other columns. So the next step is selecting some genes for visualization. Okay, so if you go to the second tab, visualization, the first one is going to show a heat map of expression. So the genes we selected versus the cell lines, the columns. It's interactive heat map, so if you pass the mouse through it, you can track the values over it. And also there is some clustering based on the gene expression. So I can cluster the samples considering all genes, okay? I could cluster by only our selected genes. So in this case I have some genes that has standard deviation 0, so you could not like cluster the samples. So let's select more genes here. Let's see if it solves my problem. Okay, so my samples were clustered based on the genes that I selected. I could also cluster the genes I selected. Okay, so which genes are more similar to the other ones. Okay, the next step would be, okay, we have our raw metadata, raw counts here, and normalized counts. We're going to do our differential expression analysis. This tab uses the package DASeq2 from Bioconductor. So you have any doubts how it works, you can read this package manual. Basically, we start by uploading the metadata from our samples. So I'm going to load this file from the Google Docs again. So I have the name of the samples in the first column, and then any other type of metadata that could be, we can perform the differential analysis. So basically here we're going to do we're going to compare different samples from different histotypes. If there is more columns, you can use them as covariates. So we'll select one of the histotypes as reference. So let's keep this one. And then we perform the analysis. So actually what DA62 is going to do is get all other histotypes and compare to HGSOC histotype. Since the DEA analysis took a while, we added the video so we don't wait too much. Basically here are, when it's over, it's going to show you the results in the table. So for each gene, with the log fold change between two groups and the p-value. You can select which analysis you want to take a look. So it's all the groups compared to our reference level. So let's say you want to see the mock versus HGSOC. So the table is going to be rendered with the results of that analysis. You can either export that table the same way you can take a look on all genes and download the table, or you can visualize the results as a volcano plot. So just set here the thresholds you want to use and the p-value cutoff. And then we're going to have an interactive volcano plot here. With the cutoffs as our dashed line here. So you can test the mouse and see which are the genes, the most relevant ones.